we can already use that in terms of single ions to think about a really complex biological issue, which is to talk about ion channels. So this is just a quick example. For some of you, you might be very familiar with ion channels. Others might not know what these are, so I'll just tell you quite briefly that ion channels are these very massive transmembrane proteins. Essentially what they are is it's a protein that spans the membrane of a cell. And what they do is they regulate the influx of ions across that cell. So the influx of ions from the outside of the cell to the inside of the cell, for example. And you can think of ion channels as being gated. By gated, it means the gate can be closed and no ions are going through, as in this case here. Or you can talk about the gate being open, and in this case, you can see that you will have an influx of ions. So ion channels are important for maintaining a voltage difference between the inside of the cell and the outside of the cell, and they're found in all sorts of cell types in your body. But if you think about where they're most prevalent, it turns out that they're most prevalent in muscle cells and also in nerve cells, so in your neurons. And essentially, what they do in neurons is they underlie those nerve impulses, or those essentially what we call electrical signaling between neurons. You might also call that the action potential of the neurons. So essentially, they regulate this action potential, and they do so by helping to establish and then control the voltage gradient within the cells. So essentially, they're establishing or controlling or changing the difference between the charge inside the cell and the charge outside the cell. And when we talk about any type of ion channel, there are just tons of different kinds of ion channels, and you can characterize them in a few different ways. So for example, you could characterize them in terms of how they're gated and basically how they open or close. That's one way to talk about different types. Another way to talk about different types is to think about which ion they're selective for. And all ion channels are selective for a single type of ion, and we can think about how that selectivity takes place, and that's where this idea of atomic radius is going to become very important. So for example, if we look at sodium channels, and sodium channels are some of the particularly prevalent ones when we're talking about neurons. If you think about the cell membrane, and this little green cartoon is me trying to show a sodium channel here, and in this case you can see that it's closed such that no ions are getting through. However, when that gate is opened, the sodium channel is now going to be incredibly selective and only let through sodium ions and no other type of ion. And this is really interesting to think about because you can imagine in our body we have concentrations of all types of ions, and specifically some seem very, very similar to each other. So we could think about comparing the potassium ion to a sodium ion. They have the same charge of +1. The only thing that's different is that they're one down on the periodic table. Potassium is down one row. So it's going to be a little bigger, but when we're thinking about size, it maybe does not seem that significant to talk about the size. But what we find out is that it is. So what happens, this is another view of a sodium channel. So this is actually looking a little bit more at the protein structure. What all of these channels have is what's called a selectivity filter. So this filter filters out the type of atom or the type of ion that's going to be allowed through. And there's two parts of the filter. First, we need to select for actual charge. So the way that it does this is the filter is actually lined with all of this negative charge. And for those of you that are more into biology or biochemistry, that's because of negatively charged amino acid residues. But all you need to think about is that it has negative charge on the inside of this pore. And what happens then is that if something has a positive charge, it's going to be stabilized to enter this pore, whereas any negative ions are going to be repelled. So that's the first step in being selective. But now, how do we differentiate between these sodium and potassium ions? And the answer is just really beautifully simple, and it's just that the pore gets really, really tiny to the point that it gets so small that all that can fit through this pore is one single atom, or excuse me, one single ion, one single sodium ion solvated by one single water molecule. And that's all that's big enough to pass through, or small enough to pass through. And if we go up even just one row on the periodic table to potassium, what we actually see is now that it's going to be too large, and in fact, a potassium solvated with one water molecule won't go through our channel.